내가 목소리가 작아서.
오케이.
이게 여기서 하면 돼요. 오케이. Okay, first of all, I would like to thank organizers for the invitation, and I will be happy to give a talk on these T hypercells, mathematical modeling and analysis of this T cell, T hypercell differentiation. And this is collaboration with Professor Hyung Ju from Konkuk University and my PhD student, Sungwon Lee, and the biology research lab in my university. Okay, so so for those who are not familiar with this biological phenomenon, so let me introduce briefly. It's okay about this immune system and asthma, which is the topic of my talk. So the immune system is a system of biological structures and its processes that protect against disease, as you know. And to function properly, immune system must distinguish viruses or infected cells from the organism's own healthy tissue. Okay. So there are some dysfunctions, by the way, for this immune system. So let me introduce three, typical standard phenotypes. The first one is the immunodeficiencies, which means the immune system is inactive or less active than normal cells. So therefore, for example, there would be the recurrent infections. The typical example is AIDS, very well-known disease. And the second phenotype is autoimmunity. And so this means somehow this refers to the failure of organism, in recognizing their own cells as self. Therefore, it leads to attacking their normal cells. So, for example, this is arthrosis. So this is a typical example of disease. And third type is a hypersensitivity. So hypersensitivity refers to undesirable reactions produced by the normal immune system, including allergies. For instance, the typical example is the atopy and asthma. So I'm going to talk about asthma today. Okay, so because there are many alert allergens in nature, the immune system does not need to attack all allergens. However, this regulated immune system responds to allergens improperly, causing inflammation or hyperresponsiveness. All right. Then, what is asthma? Okay, so asthma can be defined as a common chronic inflammatory disorder of the airways, which is associated with airway hypersensitiveness. The symptoms of asthma include wheezing, coughing, chest tightness, and shortness of breath. Okay, so let me introduce the T hypercells. Okay, so there are different kinds of immune cells, such as B cells, T cells, NK cells. But we will be focused on the T cells, especially in particular the T hypercells. Well, these T hypercells is a subgroup of white blood cells. Especially this T hypercells is called helper because they cannot directly kill infected cells. However, they help other T cells to kill the infected cells. For example, T helper cells activate, for instance, cytotoxic T cells. Okay. All right. So this is the simple diagram for T helper cells. Okay. So you can see here the naive T cell. Okay, can differentiate into different subtypes like Th1 and Th2 here. However, there is also Th17 T helper cell, which is relatively new. Okay, and as you can see, there are lots of cytokines and chemokines involved in this business, in this interactions of immune system. Okay, so our first biological model indicates the interactions between all these cells and cytokines, such as, so for instance, the naive T0 cell, right, and macrophage and Th1, and also cytokines produced by Th1 cells. So they are somehow have a similar reactions to let's say the input, and there's the other group which is Th2 related quantities. So Th2 cells and IL4, for instance, produced by Th2. Yeah, and also here INF gamma, which is one of the important ingredients. All right. So this is our first model. So we developed a mathematical model of these interactions in this immune system regarding T helper cells. So there are some representatives in our model. The first one is Th1. So Th1 cells. So the population of Th1 is denoted by the H1 here, H1. And then here, as you can see, H1 is produced mainly by this I2, which represents IL-12, the cytokines produced by Th2 cells. And then we have another, the different type of T helper cell, 2. So this is denoted by H2. It's mainly produced by I1, which is the IL4. Okay? And also here in H1, so that also activated by I2, but inhibited by I1 here. I1, as I say, IL4, yeah, concentration of IL4 cytokine. So H2 is produced by I1, but inhibited by F, which represents interferon gamma. Okay, that denotes interferon gamma. All right, and then as I said before, the naive T cell can differentiate into two types, H1 and H2 like this. So these two are correspond to these two above. Okay, so this is a simple model. Of course, so we have. Okay, this is I explained. Okay, so differentiate into this term represents a differentiation of this naive T cell into Th1 cell type, and the second term represents the differentiation of naive cells into Th2 types. All right. And then, okay, so there is a macrophage, somehow the partnership with the Th1. So they are co-localized. Okay. So the macrophage denoted by M here has a chemotactic term here. So has the term of chemotaxis. And so this F is the interferon gamma. So in other words, macrophage migrate toward Th1, using this chemotactic term here. So in other words, due to this gradient of F, okay, however, one can notice that this term is bounded. So somehow it means, although this gradient f can be very large, so that term can be bounded. But that's a reality in biology. So we use this type of thing. Yeah, a little different from the usual mathematical chemotaxis, right? So they allow that term to be unbounded. All right, and then of course the IL-4 and all the chemokines produced by Th1, either Th1 and Th2. So IL-4 here is produced by H2. In other words, Th2, and then IL-12 is produced by H1. Okay, and TNF alpha has this type of equation, but here the important thing is here alpha. This is the key control parameter in our model. So this represents the LPS dose, so or LPS input. Yeah, in experiment, in numerics, and in analysis. So this somehow produces IL-12. Okay, so somehow the IL-12 is produced by Th1 through this alpha. Okay, LPS input. But we take here the Hill type of function with the coefficient n here. Okay. So this is the simple model. Maybe not simple, but this is quite not so simple. As you say, as a mathematical perspective, this is not so easy. But anyways, this is a full model. But because yeah.
The beta term, where does that come from?
Where is beta?
Beta is your sort of your input.
Where is beta?
Previous.
Previous?
I think the one before that.
Where is beta?
In the naive.
This is just this is not input, but this is somehow it's there, kind of production, yeah. So that's not the parameter. Parameter, but it's not so important in this sense. Okay. Okay, thank you. So, but here, so what we have to focus on is this alpha, the key control parameter, and then in the experiment, so you can write control with that. That's the input, LPS. Okay. All right. So since the model is not so easy to attack, first of all, mathematically, so we first did the numerical simulations. So this is a numerical simulations, and while the numerical simulations were performed, okay. Let me first introduce this. So this is the local dynamics, yeah, the time evolution of every quantity in response to the low doses of LPS. So here the horizontal axis represents the level of the LPS, the input, the alpha, yeah, in our model, and then no, I'm sorry. This is time. Sorry, this is the horizontal axis is time. Sorry, and then this is the graph. So as you can see, so in the case of low doses of LPS, so Th2 dominant system we have. So here H2 is Th2, and also I1 represents IL-4, secreted by the Th2. So these are dominant in this case. And on the other hand, the Th1 cell population and IL-2 concentration and F and macrophage, they are all the low. So this is the Th2 dominant asthma. Okay, so we observed. On the other hand, so this represents the reaction, yeah, the immune, response reaction due to this high doses of LPS here. So here, as you can see, TH1 dominant system we have. Okay, so H1 and where is M? At M, macrophages, and IL2 and F. Yeah, all those are dominant here. Okay. On the other hand, the other subgroup are the low. Okay. So this is the in response to high doses of LPS. All right. And then, so this is the graph, okay, as a function of LPS input alpha. As you can see, the system, right, switches from TH2 dominant, yeah, H2, right, dominant to TH1 dominant system. So this is what we expected. So this kind of, so as you can see from the numerics, we can expect somehow the bistability or something like that. So so we'll see later. Okay, and this is the comparison between experimental and numerical result. So here the left hand side, the picture is the experiment. Okay, and in the right hand side is a numerical result in response to high and low LPS input. So as you can see, there are some differences, but in general they have a good agreement between them. Yeah. So this is the regulation of IL-12. Okay, and the second pictures show the population of macrophages in response to the LPS levels. So again, the left hand side is the experiment, from the biological lab, and this is the numerical result. So again, this gives a good agreement. All right. Okay, and then so we looked at the full dynamics, yeah, and so numerically we use the fractional star method. So here the original system is the reaction, diffusion, and advection. So I think it fits into the title of this workshop, reaction diffusion system. Anyway, so this is a reaction, diffusion, and advection here. So we split into 3 parts, and then we perform step by step like this. So first we solve this advection equation, over time interval del t, and then secondly we solve the diffusion equation in the same way, and third we solve the reaction equation numerically. But in solving this in the second part, first part, advection term, we use a CLOPEC. Have you heard of CLOPEC? That originally used in the problem of conservation laws, but here in the first step we use the CLOPEC method. Okay, and basically we used finite volume method in this numerics. Okay, and moreover we used the strength splitting to have second order accuracy here. So in other words, we split even the time interval like del over 2. So in this way. So to be more precisely, first we solve this chemotactic term, and then diffusion term, and then reaction term, and then again diffusion term and chemotaxis term. So in this way we can obtain second order accuracy in the numerics. Okay, this is the full dynamics. So this is the graph in response to a low dose of LPS when alpha is 0.1. So as you can see here, TH2 dominant system we can observe, and then in contrast, No. So this is intermediate dose. Then you can observe the mixed populations of TH1 and TH2 here. Okay, so they are mixed. When alpha is 10, which is high dose of LPS, so we as we can expect, we have TH1 dominant, and macrophage is co-localized with this, so therefore we have the same picture. Okay, so this is the response, the reaction according to different doses of alpha, like this. Okay. And then we also perform the sensitivity analysis, because one of the difficult part of mathematical modeling is to find and analyze parameter values. So therefore, this is very important to perform. So basically the objective of sensitivity analysis is to identify critical inputs, so say parameters or initial conditions of a model, and quantifying how input uncertainty impacts model outcome. So this is the purpose of doing this sensitivity analysis. So we performed like this, but in general it means too many simulation runs. So we decided to use PRCC. PRCC is partial rank correlation coefficient. So PRCC is a robust sensitivity measure for especially nonlinear relationships between the input and the output. So we did like this. So as you can see, this is the result of the sensitivity analysis. So as you can see here, so the alpha has a strong correlation with all the variables. So this I forgot to say the PRCC has the value between -1 and 1. So -1 means the strong relationship negatively. On the other hand, +1 means the strong positive correlation. Okay, so therefore the positive one, upper one, have positive correlation with alpha, which we expected. Okay, so -1 is the negative, okay, correlation with alpha LPS dose. So this is it means basically the negative ones are corresponding to TH2, okay, the group, and the positive ones correspond to TH1 group. Okay, so this is the result of the sensitivity analysis and all these things. Okay, so I'll skip. And then our second model, moreover, we observed, as I said, we can further grouping. Yeah, we can group into these two subgroups. So therefore, we can reduce the full model to this very simple looking model like this. Okay, so 1 and 2, basically two quantities. Yeah, we look at. So we made the mathematical model corresponding to the B, the second model. So this is basically TH2 and TGF beta. So H and G. So TH2 is produced by self-activation like this, and then inhibited by H, which is inhibited by G, which is TGF beta, and then the TGF beta is activated by G. Okay. H, TH2. So this is the very simple. And well, first we looked at local dynamics like this. And this is the time evolution, corresponding to low dose and high dose, and like this. So this is numerics again. Okay, and this is a very interesting picture showing bifurcation. So as you can see here, so we observe the bifurcation here. So as the alpha, as I say, is the parameter, our parameter, control parameter, and as alpha increases, we see we observe switches from the monostability to bistability like this. So this is a good observation for the bifurcation with respect to the alpha here. And then this is the Jacobian, so we can compute the Jacobian to study the stability and so on. And so let me show you this graphical stability analysis. As you can see, when alpha is small enough, we have just a monostability. So therefore, there is only one steady state here. So everything, right, merged to this point, stability point. And then this is the critical case. And then when alpha is large enough, we have two stable points and one unstable point. Yeah? So we can observe this bistability graphically, numerically. And then we looked at the full dynamics with the diffusion terms. And then, well, I'm going to skip this, the analysis part. So we can show the existence, and for those who are interested in the mathematics, we can show the existence and the regularity. Yeah. So I'll skip all this mathematics part. Okay. So and then this is the numerics. Again, we have the same type of result, and also we performed sensitivity analysis like this, and this is the based on these papers. Thank you. Yeah, so I'm done.